So today we'll talk about the second topic in Multisystem 2, which is adaptive immunity. So actually the adaptive immunity, it is a collection, the adaptive immunity system is a collection, actually a function of collection of specialized lymphocytes. And each cells express multiple copies of the same receptor or the same antigen receptor. And of course, these receptors are specific to a single, highly specific to a single antigen. As you know, lymphocytes are generally long-lived, and they are continuously patterning and trafficking between the lymphoid organ or the immune organs and the peripheral tissue, searching for the non-self substance to deal with these invaders. So we have, of course, the primary immune organ, and it's actually the site of the development of the lymphocyte and provide the development of the lymphocyte antigen receptor. The first primary immune organ is the bone marrow, and it is actually the main site for the original stem cells development for both B and T cells. The progenitor T cells are produced during adolescence and transferred to the thymus, while for B cell stem cell, they differentiate into mature B cells, and mature B cells is characterized by the expression of functional surface IgM antigen receptor and IgD. These mature B cells enter the circulation and traffic between different secondary immune tissue. So if we look closely to the B cell development, you see that the stem cells, the differentiation between the different stages of the B cells can be understood from the type, from two actually criteria. The type and the number of the receptors, the number of the protein that is expressed on the surface of the membrane of the cell, and as well the stage of the rearrangement that is occurring during development. So you can see the stem cell, once the mu chain and the DG gene arrangement have occurred, and the addition of the CD19 and CD22, hence the stem cell now is differentiated into pro B cell. Soon after that, the addition of CD9 and CD10, while the further rearrangement of the gene segment will result to another form of differentiation, which is a pre-B cell. Soon after that, it will reach the immature B cell, which is still existing in the bone marrow, through the addition of the CD20, 21, and 37, and the loss of CD9 and CD10. So you can see it's a kind of the process. It's kind of like the different pattern of the molecule expressed on the surface that can mark the developmental stage of the B cell. Soon after that, in the immature, you will see that the membrane now acquire or have on its surface the IgM and IgD. Once these immature B cells encounter an antigen and become mature, and under the influence of the antigen and the T cell cytokines, interleukin 2, 4, 5, and 6, it will be activated B cell, which is either differentiated into plasma B cell and start to secrete IgM antibody, and it can do as well Ig switch to other types of antibody, or form memory cells, and of course memory cells will confer a longer immunity toward the specific antigen. For the second primary lymphoid organ or primary immune organ is the thymus. And actually the thymus is the site for the T cell stem cell differentiation into mature T cell. And we can now differentiate between three major types of T cell. The helper T cell is characterized by the expressing of the co-receptor CD4 together with the T cell receptor that binds to MHC class 2, that's T helper cell. While the T cytotoxic cell, you can see that T cytotoxic cell is totally different because it has a T cell receptor that can recognize MHC1 and binds to MHC1, and another co-receptor which is CD8. While for the Treg cell, it's a third type of T cell, which is very important in regulating the immune response, and it functions like a brake or diminish the immune system, the activity of the immune system, is a regulatory T cell. And this regulatory T cell express as well CD4. If you look closely, it will look like it's a T helper cell, except that it have a very high affinity receptor for interleukin 2, which is CD25. And as well, T regulatory cells have the ability to bind to MHC class 2. For the development of the antigen receptor, so we have now immature lymphocyte that can rearrange genes within the immunoglobulin, a gene loci, which is either B cell or the TCR loci, as we said in the last lecture. And they try, both types of cells try to assemble a very unique antigen receptor. The help of the enzymes, which is recombination activating genes, which is called RAGs, these RAGs direct the process of rearrangement to produce vast variety of possible antigen receptor. There is a positive selection that screens for functional antigen receptors. The final rearrangement must pass the negative selection. And actually, the negative selection is very important to eliminate receptors that recognize self-antigens. Any failure to eliminate this self-reactive antigen receptor, of course, it will result into autoreactive cells, and it is the basis of autoimmunity. The mature naive lymphocyte, actually the naive lymphocyte, it's a term for the lymphocytes that have not yet met a specific antigen. They emerge and circulate. What is the anti-inceptory response? Actually, this response provides antigen receptors for a large variety of encounters. So our immune system can generate a huge number of cells are produced, and each with a specific antigen receptor can recognize separate and unique antigen. And the immune system try always to produce a repertoire of lymphocytes capable not only for recognize current antigen, but they are capable of recognize any new or any novel antigen that may encounter the organism during the lifetime. For the secondary immune organ, so actually these and one of the famous examples is the lymph node. It provides a niche for the mature B and T cells to be educated to respond to specific antigen. Okay, so it need to be like we say, it need to be fine-tuned, these B and T cell, to respond to specific antigen. So actually these secondary immune organs, actually it is a compartment for circulating this naive mature T cell, these cells that didn't encounter yet an antigen, to enter and screen for an antigen recognized by its own TCR. So this naive lymphocyte, never exposed to an antigen, enter these secondary immune organs. And starting the entry to the lymph node as we said from a previous lecture, from the high endothelial veneer, this naive T cell express one of the important receptor, which is CD62L, that recognizes the CD34 on the endothelial wall. So through this interaction, this T cell can enter by extravasation. The naive T cells express as well as we said, the 62L-CD, and the antigen-educated lymphocyte express the CD44 protein on its surface. This niche provides space for meeting between the antigen-presenting cells that can display the antigen recovered from any inflammatory sites. The selected T cells, why it's selected? Because it is activated by the antigen that it is expressed on the surface of the antigen-presenting cell. Proceed through a phase of proliferation and differentiation to do its job, which is the adaptive response. The differentiated T cell then guide the response of the humoral. That means it control over the B cell to guide it to do its function of secreting antibody, and as well of cell-mediated or cell-mediated if needed, which is the cell-mediated cytotoxicity. So let's see here what's happening to the T cell. So originally, as you can see, we have, hold on. So you have the bone marrow, of course, you have the stem cells, which is characterized by having a receptor, which is CD34. And this actually is a marker, T cell marker, identifying cells that will differentiate into a T cell. So upon the action of some hormone like thymic epithelial hormones, in the fetal subs, for example, the cortical thymocytes, they found that 98% of these cortical thymocytes will die in apoptosis. Why? Because they found that these T cells are unable to bind to MHC class 1 or class 2. And as you know, one of the crucial function of T cell is to bound to MHC, either MHC1 or MHC2 together with the co-receptor. So soon after that, you will see phase 1 in the differentiation, in which this thymocyte, actually we are talking about the 2% that will live. It's the start of the addition of a number of proteins on the surface of the cells, including in phase 1, CD2 and CD25 and 44. And then we have the CD1, 2, 4, 8 as well. And the loss of 44 and 25, while in phase 3, which is occurring in the medulla, started the TCR start to appear bound with the CD3 complex with the CD3 complex, and as well the loss of CD1. Soon after phase 3, before that, you need to understand that from phase 1 to phase 3, the different types of, or the different developmental stage of the T cell is characterized by different sets of receptor on its surface. So in these phases, actually the T cell acquire and lose specific membrane markers. Soon after that, the differentiated T cell will leave the thymus. 90% of these differentiated T cell will be having the alpha and beta TCR. As we said in our last lecture, we said that around 95% of the receptors on the T cell are alpha and beta. And as well, they have these sets of proteins, which is CD2, CD3, CD4, CD8. And of course, these receptors, together with the co-receptor, will respond to peptides of the antigen bound to MHC. While the other percentage, few percentage, 4% or 5%, as you said earlier, sorry. So it's for the gamma and delta TCR, and it have the CD3 receptor. So you can see that this type of receptor will respond to non-peptide antigen, and they lacks the MHC restriction. So actually they are antibacterial, they have antibacterial properties, and they have increased cell-mediated immunity against viruses. To continue this, you will see that after phase 3, as we see, we have once this differentiated cell after phase 3 leave the thymus, as we said, it have these two routes of either expressing the receptor of alpha and beta or expressing a receptor TCR of gamma and delta. So you can see the major difference here, that for the first one, you have the CD4 plus, CD8 plus, which is lacking here. If you took the branch of the cells that express the alpha T, you can see that 70 or almost 25% of these will acquire only the CD8 and will able to differentiate into two important cells, which is the TC cells and the T suppressor cells, while the other 70% will express the CD4 protein or co-receptor, and usually it turns THO, which actually the precursor of different type of T helper cells. And you can see here, you can see three different types of T helper cells: T helper 2, T helper 1, T helper 17, and actually you can distinguish between these three different types in the type of the cytokine they secrete. And each of them is involved in a different function. As well, for T helper 17, you can see that it can be differentiated into Treg cells, which start to secrete the interleukin 10 and TGF beta. And if you remember, we said that Treg it can regulate the immune system to avoid the overwhelming action of the immune system. And this Treg, it provides antigen-specific regulation and tolerance as well. For the three types of the hold on. For these three types of the For these three types, T helper 1 and T helper 2 and T helper 17, you can see that they have different functions. For the T helper 17, most of the function is related to the inflammation, while for the T helper 1, you can see that it activates the B cells, and here comes the interaction as well for T helper 2, and here comes the interaction between the B cell activation and the T cell activation, and as well it can increase the cell-mediated immunity as well. It relates to the antibody secretion. For the T helper 2, it has a role in the B cell differentiation and the proliferation. It can activate eosinophils. It can activate class switching and increase the amount of antibody, and as well downregulate the T helper 1 function. So in these two figures, we have seen the T cell development and the B cell development originally. So we can see here the different types of T cells that can be differentiated from the T helper O under the influence of different cytokines. So you can see here, and actually collectively, they are called the family of helper T cells. So you can understand that there is a lot of different types of T helper cells. So initially you have the naive T cells that upon activation by the antigen represented by the dendritic cells, it differentiates into initially to T helper cells. And T helper cells start to secrete interleukin 2 for autostimulation and basically to proliferate in the form of T helper cell. In the presence of the interferon gamma, together with this interleukin 12, this T helper 1, O can be differentiated into T helper 1 that can secrete interferon gamma as well. You can see as well that among the different other cytokines, you can see different types of T helper can be differentiated from the T helper O. And each of them can secrete its own specific interleukin. And you can see sometimes the interleukin that is secreted by the differentiated, sometimes the differentiated T helper, terminal T helper cell can be the same as the cytokine that lead to its activation. So for example, you can see that T helper 2 can secrete interleukin 4 that is actually used to differentiate the T helper 2 from the T helper O. Others differ, like T helper 9, it secretes interleukin 9 and interleukin 10, but they are stimulated to differentiate by interleukin 4 and TGF beta. Other types of cell, you can see other types of cells like the Treg, which can differentiate and start to secrete interleukin-10 upon the activation by the retinoic acid and TGF-beta. And as well, Treg, the Treg and the T helper 3, other types of the of the different differentiated T helper cells upon upon the activation of other cytokines. So here you can see the family of the helper T cells, and all of them trying to suit the different threats to the immune system. And as I said, one of the important cells is the Treg, because Treg actually works as a regulatory cell for the overwhelming to control the overwhelming of the immune system. So we'll talk now about the Germinal Center. So what's our Germinal Center? Actually, these are regions inside the secondary lymphoid organs or second immune tissue. And it's a niche for the B cell actually to differentiate and expand and differentiate to start secreting the antibody with different diversity and specificity. And actually, it consists of these germinal center consists of two regions: a dark zone and a light zone. And dark zone is characterized by containing the centroplast, while the light zone is characterized by containing the centrocytes. And actually, the centroplast and the centrocytes, it's other nomenclature for the B cells based on the region they occupy in the germinal center. So the dark zone, actually, it is the site of the active proliferation and somatic hypermutation. So somatic hypermutation occurs in the dark zone. For the B cells, which is in the dark zone, these B cells in the dark zone is called centroplasts. As we said before, the somatic hypermutation can produce an enormous amount or enormous diversity of antibody. To clarify more about the somatic hypermutation, it's actually a series of point mutation that randomly alters the amino acid sequence in the antibody paratope, which is the region which will bind to the antigen. It can occur by deamination of C2U, driven by an enzyme called activation-induced deaminase, EID. The uracil is removed by uracil DNA glycosylase, and this will lead to an error that can be repaired by the DNA polymerase. And you can see from the figure the consequence of the somatic hypermutation. So originally you have a B cell which have a basic receptor on its surface, and you can see that a series of mutation can determine the affinity of the different somatic hypermutation can dictate the affinity and determine the affinity of this receptor to a specific antigen. So you can see in this type of cell, this mutation lead to not a good affinity and a poor survival. So this cell will die eventually. While the last one, you can see that the mutation that occurred resulted into a very high affinity toward its specific antigen. And in this case, the survival of this kind of cell or this clone of the original B cell will be very good. For the B cell, it'll do something called editing for the immunoglobulin to make it very high affinity very higher in affinity to interaction with antigen. So you can see here from the figure as well how the mutation or the somatic mutation can result to higher antibody antigen affinity. So you can see here, for example, you have an early primary response to the antigen. You can see that there is two mutation occur, and this resulted to this kind of affinity between the antigen and antibody. So like if a series of other mutation, you can see in a tertiary response toward the antigen, the number of mutation have increased, and the efficiency of this mutation led to a very high affinity between the antigen and antibody. So you can understand here how these mutations is very important in increasing the affinity of antigen to antibody. And even you can understand now that the response of the B cells towards an antigen, it differs dramatically between the early response and the late response. And this explains why our body immune system respond very efficiently on subsequent encounters of the same antigen, because it manages over time to do the correct number of mutation and correct mutation actually to lead to very high affinity between the antigen and antibody. So we have now, in the Germinal Center as well, we have epithelial-derived follicular dendritic cells. And actually, these are the antigen-presenting cells. And they can bound to the antigen as well. So these FDC, or the follicular dendritic cells, actually originate, as we said earlier in the lectures, from the inflammatory side, because they can collect these antigen and from these inflammatory sites and actually recruit and present these antigen to the T cells inside the secondary immune tissue. So in this germinal center, the B and T cells actually, because they are naive, start to screen for the antigen on these antigen-presenting cells of the FDC. The B cells, which are actually the centrocytes, and T cells remain associated with this antigen on the FDC. And the B cells shift antigen recognition, as we said, by somatic hypermutation. They start to shift until the association with the FDLC is lost or the T cell intervenes. That's mean it have managed to do the correct somatic mutation to bound to the antigen. Soon after that, the centrocytes, which is the B cells, proceed through affinity maturation, while somatic hypermutation is sustained by T cell interventions. The B cell also can also change isotype, what we say the class switching, to enhance antibody function within different tissue. So the B cell, instead of secreting IgM or IgD, it can do switching to suit and to provide immunity in different tissue, like class switch to IgG to do in the circulation, or IgA to have antibody in the mucosal tissue. This isotype switching is driven as well by the neighboring T cells. So let's understand more clearly in this figure. So as we said, the germinal center have two regions. The dark zone, as you can see, which contain a B cell, which is termed at that position centroblast. And you have this other region is called the light zone. So originally, this B cell that came originally from the bone marrow start to do reconstructing IgG specificity by somatic hypermutation. You can see that it can migrate, this centroblast, it migrate to the light zone, and hence it is named centrocyte. And it interacts with the FDC, or the antigen-presenting cells, which is presenting the antigen, together with interaction with the T cell. You can see the helper T cell here is around the antigen-presenting cell. The interaction of these centrocytes with the antigen-presenting cell, together with the T cell, you can see that it will lead to activation of these centrocyte to do either start to further differentiate or do Ig class switching, of course, under the guide of the T cell cytokines. So once it recognizes the antigen very well from the antigen-presenting cells by the help of the cytokines released by the T cell, you can see that cells that fail to have a high affinity after doing successive multiple rounds of somatic hypermutation, if they fail, this B cell, if they fail to have a high affinity receptor for the antigen, they soon undergo apoptosis and dies, these centrocyte or these differentiated B cells. In the other case, if it succeeds to form a receptor with a high affinity of antigen, it will give an it will be differentiated into a plasma cell, which can be which is a secreting form of the B cell and start to secrete the antibody specific of for the antigen that have been recognized on the by the dendritic cells. As well, you can see that soon after that, the other branch of differentiation of the centrocytes is to form a memory B cells, and this, of course, will give longer last immunity. So here you can see how the class switching occur, and what is requiring a cytokine to do the class switching. So originally, you can see that of course, the color is important. So you can see the color coding. So if you go back to the figure, you can see that there is a difference between the centroplast, which is appear like light blue, and the centrocytes, which appear like pinkish in color. So you can notice the difference between the centroplast and the centrocyte. So here you can see, of course, this will be the centroplast, and upon the activation of interleukin-2, it can differentiate to centrocyte that with further activation of either interleukin-2, interferon gamma, interleukin-4, or TGF-beta, it can differentiate into different type of plasma cell that is capable to secrete different type of antigen to suit different functions of the immune system. And this process, you can see it is a class switching. So in this case, you can see that the B cell, which is originally express IgM on its surface or something like that, or IgD, originally can now do class switching upon the activation of these upon the activation by these cytokines. So let's talk about the lymphocytes. So as we said before, we have lymphocytes that have the T cell, especially that have alpha and beta TCR, which represent 95%. Actually, it's found on the thymus-derived T cell and can recognize peptides presented by MHC class 2 molecules. If you remember about gamma, and some of you asked about the gamma delta TCR, is found on the mucosal epithelial T cell, and they can recognize if free peptide and doesn't need antigen presentation. And that's what I have said before, that you can understand that the T cell, the type of T cell which have a gamma delta TCR on its surface, it provides an immunity as well, but with a less specific immunity, because it doesn't need any antigen presentation. So we have the antibody, as we said, are produced by the B cells. You have generally you have two types of B cell. You have a B1 cells, which is found in the mucosal area and produce antibody to polysaccharides. And these B1 cells, B1 cell immunoglobulins, have a limited repertoire to different antigen. So they don't have a diversity in their specificity. While the B2 cells are produced in the germinal center. So what we have just said about the centroblast and centrocytes, all of them collectively called the second category of the B cell, which is the B2 cells. And they have the ability of doing a lot of rearrangement, somatic hypermutation, and the class switching as well. The B2 immunoglobulins, this means the antibody which is secreted from the B2 cell types, have virtually limitless different diversity of antigen recognition ability, and they can differentiate into plasma cell. So we need to differentiate here between the type of antibody which is secreted between two general classes of B cells. The B1 cells provide antibody which have limited specificity since they only recognize polysaccharides. So what about the mature DC or the mature dendritic cell? That actually the main cell that activate T cell actually. Initially, as we said earlier in the lectures, these immature dendritic cells accumulate and identify antigen at the site of inflammation. Due to the exposure to the PAMs, induce its maturation and direct its migration, if you remember, toward the lymph node, which is the secondary lymphoid organs or second immune system, immune tissue. These mature dendritic cells, as we said, present the antigen to the T helper cell using the class 2 MHC molecules in the lymph node, and they take residence in the lymph node adjacent to the mature naive cells and mature naive T cell. And as we said, the mature naive T cell is still still not active. Okay, so soon after that, the mature dendritic cells present these antigen, and to try to educate these naive T cells, and as well express CD8086 and increase the surface display of MHC class 2 bound antigen, because as we said, it's a restriction for the T helper cell to identify the antigen presented on the antigen presenting cell together with class 2 MHC. And this antigen specific T cell begin after that, after educated by the mature dendritic cell to start development and the clonal expansion. While the naive B cell bind the antigen in the draining lymph node as well, and the antigen can be internalized and expressed on class MHC class 2. And this specific T cell as well recognize this presented antigen, and its main step in the activation and the stimulation of B cell differentiation and activation. So what about the specialized lymphocyte? There are a set of cells that are specialized lymphocyte. They are generally localized in the mucosal boundaries and express low copy number of antigen receptor. One of them is the B1 receptor, we have just said that. One of them, the secrete antibody, which is specific to carbohydrates, as we said before, polysaccharides, carbohydrates and lipopolysaccharide. We have as well marginal zone B cells, which is located in the spleen, and they as well produce antibody to polysaccharide. And we have as well the cell that have been asked before, the gamma delta T cell, and as we said, they express an alternative T cell receptor because it's a gamma delta, represent 5% of the total cells that have receptors, are located, as we said, at the boundary areas of gut mucosa and the epithelium. The fifth type of the specialized lymphocyte is the NK cells, which is actually search for host cells that have a bearing, because as we said before, NK cells is responsible for attacking the virus infected cells or the cancer cells. If you remember the video we have seen earlier in the lecture. So these NK cells search for hosts that have a class 1, because as we said before, this class 1 MHC is almost represented on all nucleated cells. So if there is apparent or abnormal expression of this MHC class 1, it's got identified by NK cells, and these NK cells start to do cell-mediated cytotoxicity toward these kind of cells. So this NK cell recognizes the target cell based on the antibody labeling, complement labeling, and the screening for low MHC class 1 expression. So even if it's not abnormal, but even if this normal cell express very low frequency of this type of molecule. Of course, the NK cell express receptor for complement like C3b, C3a, C5a, and as well for IgA and IgG. And NK cells engage a target cell through the lectin activity. If you remember the lectin, that this is a pathway that leads to activation of the complement system, or by recognition, if the target cell is recognized by and labeled by antibody and or complement. That's why NK cells express receptor for complement and antibody as well, because they recognize target cell labeled with antibody or complement. These NK killer receptors screen for the absence of MHC class 1 using an inhibitory receptor. And this engagement to the killer inhibitory receptor deactivate the NK cells. So one of these, actually one of the main mechanisms of cancer cells to inhibit the NK cells through the expression of a killer inhibitory receptor, which is called KIR. So once they express this kind of receptor and the NK cells detect this kind of receptor, it will be deactivated. The NK cells kill target cells by inducing apoptosis if the KI receptor is not triggered. The NK cells engagement activate a cytolytic mechanism, as we know before, and this target cell death by apoptosis is driven by granule exocytosis or TNF pathway. If you remember from the animation we have seen before in lectures, we have seen that NK cells can attach to the target cell and start to secrete the some of its granules inside the target cell to degrade it. Again, for the killer inhibitory receptor, this killer inhibitory receptor actually is located on the surface of the NK cells. Once it is not triggered, the NK cells can be active and kill any altered cell. For the natural killer T cell, which is actually which is not the NK cells, it's actually share properties to NK and T cells. They display alpha beta TCR. They can recognize glycolipid displayed by CD1. They display a killer cell lectin receptor, which is called NK1.1. They can produce interferon gamma. Another type of specialized lymphocyte is the plasmacytoid cells, and it's actually low abundant leukocyte, less than 1% of the circulating leukocytes. It is lymphocyte derived, do not express any complement receptor like CD110, do not express CD14, as it is not responsive to liposaccharide. So you can understand that CD14 is involved to the responsiveness to lipopolysaccharides. They can express toll-like receptor 7 to detect single-stranded RNA, and actually you can understand that these type of cells can recognize viruses, because viruses, some of the viruses, example the coronavirus, have a single-stranded RNA. And as well, it can express toll-like receptor 9 to detect a CBG island. And what is CBG island? Actually, it's a region of DNA that is consists of CG, CG, CG. So basically, it can recognize DNA if it's introduced inside our body. These type of cells as well can produce large amount of interferon alpha and beta, and this type of cell is crucial for to defend our body against the viral infections. So here in this figure, you can see how the different axes of the immune system is like a summary, a simple summary for the interaction of different branches or different axes of the immune system. So initially you have, for example, the virus. You can see the virus. It can be recognized by the dendritic cell. It can be recognized by the dendritic cells or the macrophage, and this and it can be internalized, and the viral antigen or parts of this viral are displayed on the surface of the antigen-presenting cell, the dendritic or the macrophage cells. You can see it have internalized the virus, processed its protein code, and it can express on the surface the viral antigen. Soon after that, this viral antigen, you can see now, it's even an infected cell, virus infected cell. Soon after that, this one is, you can see that the antigen presenting cells have activated T helper cell. You can see now, because T helper cell by its T cell receptor can bind to the viral antigen which is expressed on the antigen-presenting cell, which lead to secreting of cytokines that activate the cytotoxic T lymphocyte to bind to the infected cells and do what it do with cell-mediated immunity. Soon after identification of this one, it can differentiate into a memory cytotoxic T cell and cytotoxic TC cells as well to continue killing other infected cells. And the cytokines, which is released from the helper T cells, can stimulate as well to form the memory helper T cell. So you can understand now that the memory cells actually arise from every single type of the lymphocyte. So the cytokines released upon the activation of the T helper cell either activate the cytotoxic T lymphocyte or do activation for further differentiation of the helper T cell to form a memory helper T cell. Soon after that, of course, these memory helper cells, memory cells, either T cytotoxic or T helper, will confer immunity for subsequent exposure to the same microbe. You can see as well that the cytokines that is secreted by the activated T helper cell can activate the B cell as well to differentiate into the two major types, which is actually the memory B cell and the plasma cell, to secrete antibody toward as well. It can be secreted toward the virus as well. So you can see here how the different branches or axis of the immune system can cooperate for combating or defending the body against the virus. So actually this table lists the different types of the T cells family or different types, and the symbol they use to identify these different T cell subset, and actually the function and the type of the cytokine they secrete. Okay, so for example, let's say, and actually we said most of these cell types during the lecture. So for example, you can see the T helper 1, actually its function, it's intracellular bacteria. It is against intracellular bacteria and virus, and this actually under the action of the majorly the interferon gamma. So you can see that one of the other examples, the cytotoxic T cell, represented by TC, and actually it is involved in cell-mediated immunity, and actually it do its action by using one type of the a type of the granules, which is called peripherin granzyme, giving the impression it can form pores inside the pores on the membrane of the target cell. You can see as well the activated helper T cell and the naive T helper cell. And you can see that both of them, although they have the same symbol, but one of them is quiescent and the other is not quiescent because it's activated by the, as well, if you remember, by the interleukin 2 as well. And you can see the only difference between these two cells, that this one express either CD4 or CD8, okay? And you can see as well the difference between the natural killer cell and the natural killer T cells. And actually one of the major difference between both of these type of cells, the type of the secretory cytokines or the enzyme they secrete in order to have their parts in the immune system. And this concludes our lecture today for the adaptive immune response.